Hi, welcome to MTE 2023. So let's find out a new product from University Selangor, an innovation for the purpose of monitoring Pterygonochitis pardalis called Pleco. It is an eDNA-based identification assay for rapid monitoring of the passive aquatic monster Pterygonochitis pardalis, or commonly known as the Amazon sailfin catfish. Ladies and gentlemen, did you know that Malaysia is one of the world's megadiverse countries, ranking 12 in the World Biodiversity Index? As a result, Malaysia Biodiversity Information System (MyBIS) has been developed as a one-stop repository for biodiversity information. MyBIS is a platform for the Global Convention on Biological Diversity members to exchange biodiversity information. However, according to the present assessment, 48% of all known species in Malaysia, which include 25 families, 28 genera, and 133 species, are threatened, with freshwater fishes having the largest rate of vulnerable species, which account 87%. Multiple studies have determined that freshwater species are extremely vulnerable, with habitat loss accounting for 78% of the reasons, overfishing accounting for 27%, and bycatch accounting for 23%. In addition, the introductions of non-native species into our freshwater ecosystem can have detrimental effects on native fish populations. Invasive species can outcompete native species for resources, prey on native fish, introduce diseases, and alter the habitat structure, which leads to declines or even extinctions of indigenous fish species. Invasive fish species have become the main reason for the loss of biodiversity in aquatic habitats and have numerous effects on native fish and aquatic systems not only to Malaysia but globally. Not only have effects ecologically, the presence of invasive species can have various effects on human health, both direct and indirectly. Pterygonochitis pardalis is one of the most threatening invasive species which has been deliberately introduced into our ecosystem. The introductions of the Amazon sailfin catfishes into natural drainage of Southeast Asia in 1980 to 1990 has led to their noticeable naturalization. The occurrence has spread particularly to Malaysia, Philippines, Singapore, and Taiwan. And in several research papers, there are two species of Pterygonochitis were reported found in Malaysia. The populations of P. pardalis were found in Langat River Selangor, Perak River, Perak, Muar River, Negeri Sembilan, and Gombak River Selangor. The assessment of fish biodiversity in freshwater environments of Malaysia is often challenging. Detection probabilities for invasive species are typically low in all environments, particularly in aquatic environments where organisms are hidden beneath the water surface. On top of that, conventional sampling methods require long-term surveys, which labor and time-intensive. In Malaysia, traditional detection methods such as manual search and trapping are widely used, although it is still a reliable method. It is time-consuming and requires extensive monitoring and sampling. Therefore, development of Pleco as a biomonitoring kit for this species to environmental DNA will aid in monitoring system of this species. The ability of environmental DNA, also known as eDNA, in order to identify the presence of organisms in small volume of water has been realized over the last decade. It's a new sensitive technology for detecting the presence of organisms and their dispersion using DNA left behind in the environment, such as in water or on land. This method allows for far larger species distribution surveys, as it requires significantly less time and resources than traditional methods. Pleco emphasized the possibility of being a novel species-specific assay for Pterygolechitis pardalistes, one of the most impactful invasive species in Malaysia. This provides a tool for rapid observation and quantitative monitoring for the invasive species. It's rapid and reliable. This innovation was created to address three primary issues. Firstly, to identify diagnostic and suitable gene fragment for species-specific identifications of Pterygolechitis pardalistes. Secondly, is to develop assay to distinguish different species of Pterygolechitis and aid in correct identifications of the species level. And thirdly, is to rapidly detect Pterygolechitis pardalistes DNA at a very diluted concentration in environmental samples. These innovations offers a rapid solution. What is needed is just water sample from the environment. Once the water is filtered, tada! eDNA will be extracted from the water sample. And using our eDNA assay, presence of Pterygolechitis pardalistes can be detected within only 2 hours. The key benefit of our innovation is a rapid response time. Researchers and even volunteers can now assist in the quick detection and monitoring of Pterygolechitis pardalistes. Well, is the product ready? Pleco Quest was tested in a real-world setting and yielded positive results. Is this product has commercial potential? Invasive fish species have become the main reason for the loss of biodiversity in aquatic habitats and have numerous effects on native fish and aquatic systems, not only to Malaysia but globally. So as a result, it is imperative that the occurrence of invasive species be monitored using advanced technology in order to prevent additional loss of native species and meet future fish demand. And meet our team, a collaboration of researchers from various universities who are passionate enough to eradicate this passive aquatic monster. This is also a joint effort with research personnel from Malaysia's Fisheries Research Department who directed the research team with information on the occurrence of Pterygolechitis pardalistes. And this project has also received a media coverage by the Symphony Alam RTM Center. This study, funded by the Fundamental Research Grant Scheme, discovered that eDNA technology can be used to expand biomonitoring diagnostic capability. Using biotechnology, this assay innovation allows us to move detection work from the lab to the field. We hope that this innovation will pave the way for future biomonitoring approaches that are also sustainable for our biodiversity. With all that,